Good afternoon. I'm Robert Keeval, the CEO of IMMR's US headquarters in Minneapolis, and I am joined by my colleague in Paris, Dr. Luc Baer, a veterinary surgeon who is one of IMMR's founding partners and senior scientific directors. Our presentation will highlight the critical role of preclinical science in healthcare innovation, and our focus for today will be the subspecialty area of vascular intervention. In the development of new medical innovations, the preclinical phase serves to inform and help perfect the design of new products and technologies and to demonstrate their suitability for advancement into human clinical evaluation. Generally speaking, the elements of preclinical science include preclinical studies, pathology evaluation, and the training of clinical investigators on how to use new products and technologies safely and correctly in human patients. The preclinical phase is a significantly value-enhancing stage of product development. The earliest preclinical studies serve to help validate the basic proof of concept and procedural safety of a new product or technology and help to inform initial elements of product design. Ongoing feasibility and R&D studies help perfect the design of new products through iteration of prototypes, as well as any associated implant procedure and delivery tool, and begin to shed light on the potential clinical utility of new products. And the final GLP compliance studies serve to document the chronic safety and performance of new products and technologies to help justify their approval to proceed into human clinical evaluation. Now, the results of preclinical studies are of vital importance to management teams, of course, but this is hardly just an inwardly facing activity. There are important external stakeholders as well. For example, regulatory agencies and institutional ethics committees that need to provide approvals for new products to be used in human subjects, thought leaders and investigators who need to be willing to support and utilize new technologies in human patients, and of course, funding agencies and investors that decide which programs receive funding, and if so, at what valuation. The field of vascular intervention involves treating disorders of or related to blood vessels in the heart, the periphery, and the central nervous system. The list of vascular intervention technologies is long and diverse, and it's growing every day. And this can represent a challenge to both caregivers and regulatory agencies who work to keep up with the pace of innovation. And as technologies grow in sophistication, so too does the complexity of their development pathway. With this in mind, I'll turn to Luc to continue the discussion.
Thank you, Rob, for the introduction. I'm Luc Baer, and I'm talking here from IMMR in Paris. And good morning, good afternoon, or good evening to everybody. I'm one of the co-founders of the Research Center and also scientific director here at the lab. My part of the presentation will focus on the preclinical pathway. And on this slide, you will see the different steps you have to go through to develop and validate your medical device. So you start first with proof of concept until you achieve first in man. The proof of concept phase is the very early phase where actually your prototype will meet reality. When I say will meet reality, it's first in vivo implantation. And these prototypes that our partners come with when they come and meet us initially are the fruit of the brainstorming between physicians, engineers, and maybe already included regulatory people that bring to us an early technology that we will test in the proof of concept. When the proof of concept is achieved, you will turn into the feasibility and R&D study. The feasibility and R&D study is the part of the preclinical pathway that will enable full validation, and testing of different types of prototypes. Here you see on my picture that I represent a loop. I call it a beneficial feedback loop from the ground. And the ground is actually the testing in the animal. You come with an initial product, an initial prototype, and you implant it in a series of animals. You might encounter failure. And here you are expected to encounter failure. And this is here to help you improving your devices. If the device is not fully efficient or safe, you go back to engineering, you redesign your device, and you come back with an improved device that, again, we will test in a series of animals, and so on until you have a refined, safe, and reproducibly efficient prototype of your device. Then only you can enter the regulatory study phase of your development. And this regulatory study phase is performed by evaluating the final frozen version of the device. Why do I insist on the fact that this should be the final frozen version? Is because in this phase, the regulatory agency will not like or will not accept that modification should be made to the device that is tested for regulatory application. And then, before going into humans, you have to perform a stringently designed and documented training of the initial clinical team that will implant your device in humans. It is of importance that these physicians are properly trained for patient safety, of course, but also for the success of your device in clinics and in the market then after. On the next slides, I will give you more details about the different phases I've just talked about. The proof of concept is, to me, an essential starting point. Why do I say essential? Is because if the proof of concept is not properly performed, it might not give confidence to the engineers on the scientific side, but also to the shareholders that this project has a future and that this device should be a device in which we can invest for the future. So it is of importance that it is perfectly mastered. And in such the phase of the study, you should include approximately 5 to 10 animals. And these 5 to 10 animals should serve in, for an example, an acute session to demonstrate initial efficacy and initial safety. It's not going to be the full demonstration of it. It's initial results that should be positive and that should encourage the engineers and the investors to continue this project. To do so, you usually implant your first iteration of the device into healthy animals, but you should also consider implanting it into pathological models. Why is this of importance? It's because to give confidence that the concept might improve and might help for a pathological application in humans, these pathological models are then very important. And for an example, in the vascular field, we can provide you with, and these are generally accepted and designed and validated models of deep venous thrombosis, for an example, for thrombectomy devices, arterial tortuosity, that can be a challenging environment to test new wires or new catheters, soft and hard clot obstruction models, because as you can imagine, it can be more difficult to prove that your wire is efficient going through clots when these clots are chronic clots, and in patients, this will be the case because they might suffer from their obstruction for days or even weeks. And another type of model, for an example, could be aneurysm examples of pathological models. The key challenges here in the proof of concept study is that the components and the prototype will be unrefined raw versions of your prototype. And these might be sometimes difficult to implant just because these are raw, early, rapid prototyping devices. And for that, the team you will partner with to perform this animal proof of concept should be an expert team that has a lot of experience in this particular vascular prototype or vascular device you are developing, because at this stage, the team needs to be adaptable, needs to be creative, and needs to have a mindset that is orientated towards real-time problem solving, because there might be problems and you need to solve them and be creative to be successfully implanting this device to go over the proof of concept phase. The next phase, which is after the proof of concept, is the core and the body of what you will have to perform in the preclinical phase. This phase is the feasibility study, in which you will come with several prototypes. Each time the prototype has to be tested in approximately 4 to 6 cases, because when deciding if a prototype can go through or needs to be further developed, you have to base your conclusions on solid scientific data, and you cannot decide of the life or death of a prototype only on 1 or 2 cases. That's why we strongly advise that these should be series of approximately 4 or 6 cases. Then after, only you will be able to freeze the design of your prototype. And designing and freezing the final version is very important. That's why series have to be of a certain amount of number of animals. Key challenges here is that, of course, you may require multiple iterations, but that's what it is expected. You are not expected to be successful after first iteration. It will be multiple iterations. And this is important for the engineers to understand that this phase is there to fail and to experience problems, but then to solve them. But it is also important for the investors to understand that this phase might be a lot of adverse events during this phase and multiple iterations. And the investors have to accept that this is part of the process. The next step is when the device is fully refined, entering its final version, you will enter the regulatory phase of the studies. Of course, when you talk about regulatory, you directly think about the different agency that will review your results and give you approval. And we are talking here about compliance to the agency requirements, being in Europe for CE marking, in the USA with the Food and Drug Administration clearance, or for example in China with the NMPA, the National Medical Products Administration, to give you product to put your devices in humans in China. This is the regulatory environment per se. There is another regulatory compliance that you have to seek and to look for, is that the partner you will perform the animal testing with has to comply with the highest international standards. And here at IMMR, we are proud to be part of the ALAC Association and fully accredited for our program of animal welfare. The regulatory study is usually designed to meet scientific aspects of the regulatory requirements, and also these have to be performed into a quality assurance environment that meets the requirements of the agencies. So talking about quality science within quality assurance environments. And quality science is our mission in the research center. We are here to design with you the perfect study that matches the risk analysis of your specific vascular implant, for an example. And to do so, we will design a study in which we will design test groups and control groups. The test group is also the groups in which we will implant your prototype. And the control groups are the groups to which we will compare the results of your own device. A control is defined as a device that is comparable to yours, but is already on the market and already in patients, having proven safe and efficacious in the patient applications. And that is of utmost importance that in your regulatory study, you put a control group. The time point at which we will be evaluating your device can be from early time points to very long time points according to the risk analysis of your vascular or other types of application and device. You have to keep in mind that the agency look at acute, subacute, subchronic, and chronic safety and performance of the device. When we talk about acute or subacute, we are talking about 1 week to 1 month. And when we talk about chronic, we are talking about several months up to 6 months, and more, even for longer requirements from the regulatory agencies, especially if you're looking at the development of fully absorbable devices and fully absorbable platform or scaffolds in the field of absorbable stents, for example, absorbable occluders, for example. There, the agency more and more require that a group of animals has to be implanted until you demonstrate the full absorption of your device. And that can take months or even years in the case of certain devices. The key outcomes of the regulatory studies, of course, is chronic safety and performance. And then the complete set of data has to be reported in the final report and submitted to the agency. It has to be a rigorous study planning, and then we have to stick to the protocol itself. We cannot make modifications once entering the regulatory studies. Of course, we can adapt, but major modification cannot be performed. And the key lab competency in the lab that you will be partnering with to do the regulatory study is the capabilities of doing a proper pre-screening of the animal and finding the proper model to evaluate your device in conditions that will reflect the use in humans. The team inside the preclinical laboratory has to have an intimate knowledge of your technology. They have to be experts in the technology you are developing. And of course, they have to have in-house expertise in the regulatory specific testing that needs to be performed. Here I have an example on this slide for vascular applications and for specific thrombogenicity testing. These thrombogenicity testing are mandatory for vascular devices and, for an example, for wires or catheter. And I have a focus here on two examples. It would be the NAVI testing or the AV testing. The NAVI testing stands for non-anticoagulated venous implant, as the AV testing stands for anticoagulated vascular implants. Why do I put the stress on this testing? Is because lately the agencies for vascular implants do require this testing, and even a lot more, dedicated to the risk of thrombogenicity of the devices. Then, after the full regulatory approval, we are all happy to help you training your future clinical investigation team. And for that, we train physicians before the human use to assure that these physicians will be performing the implantation with the highest safety for the patients, but also with fully secured success of the implantation for the success of your device and the market access of your device. And this is being done in animal settings and in laboratory settings that are fully comparable to the clinical use. And we like to say that the clinical platform has to give confidence to the physicians. And we like to say that the physician needs to be feeling like home. That's why the technical platform here at IMMR are cutting edge, late technology platforms. I have a final focus here on a specific type of evaluation, which is the pathology evaluation of your medical device. Why do I put a focus on pathology? Because as everybody say, and as the agencies like to say, pathologists rule the world. It is indeed a key element of the evaluation of your device. Pathology is a gold standard evaluation method of the performance and safety of your device. It is mandatory in all regulatory studies for the local tissue and the systemic effects, but it is also very informative in the early stage of animal trials, meaning in the proof of concept and in the regulatory phase, but also in the feasibility phase. It brings early insights into the body reaction to your device that has been implanted. And the aim of the pathologist is to evaluate, describe, illustrate, and conclude on the tissue reaction. The pathology evaluation includes two steps. First is macroscopic evaluation, and second will be microscopic evaluation. Macroscopic evaluation is, of course, being done with your naked eyes, looking at the tissue during necropsy and describing what the findings are. And then all these samples will be evaluated under the microscope, which is going to be the microscopic evaluation. For the macro evaluation, here the slides gives you some of the evaluation we are performing here at IMMR. It is the full inspection of the device and the surrounding tissue, looking at the implant reaction and position, the tissue healing and ingrowth around the device, fibrin deposits, mineral deposits, fractures, etc., etc. It is interesting that the macroscopic evaluation can give a full report and can give early data of interest, because at this stage, you can already have some insight and some conclusions on the efficacy and safety of your device. And it might be very interesting to have this early report for the engineers, but also for the management team to make decisions to further develop one prototype or the other. The macroscopic evaluation, of course, includes not only the device, as I was mentioning, but also the full body reaction, which goes through evaluation of each important and target organs in the body: liver, kidney, spleen, lungs, etc., etc. Some technical aspect of the microscopic evaluation. You have to imagine, and here I have the example of a vascular plug on the bottom right picture. You have to imagine that this device has subparts and that the pathologist has to evaluate the different subparts. That means we have to cut, trim, and stain tissue according to predetermined sections. And for that, you have to partner with the team that has a lot of expertise into designing the protocol for histopathology evaluation. All that is being handmade. It's handcrafted. So all that is within the knowledge and the capabilities of the team of the laboratory you will be performing the pathology with. And we like to say that we transform a 2D cutting, trimming into a 3D conclusion and interpretation. The slicing, the resin embedding, and the sectioning, the grinding, and the staining are very technical, and we will take charge of that so that in the end, what you will get is a full report with the evaluation of the local tolerance, the systemic effects, and these conclusions will be performed by our pathologists, which are trained, board-certified veterinary pathologists as the agency require for applications in this preclinical animal evaluation. And the conclusions will be drawn from qualitative, semi-quantitative, and quantitative evaluation. Here I give you the example of a quantitative histomorphometric analysis in which actually real measures and real values will enable the pathologist to give full conclusion on the potential and the safety and performance of a device when compared, for an example, to the data drawn from a control article. As you have most probably understood by now, the preclinical pathway gives you critical information about different parts of your medical device. And as I like to say to all the partners that come to us, you are not only developing an implant, you are not only developing a delivery system, you are also at the same time developing the imaging procedure to implant your device, the surgical or the interventional procedure. And at the same time, during the preclinical phase, you will be also developing the sizing algorithm, making sure that you will properly choose the size of device that needs to be implanted in the right size of anatomy, so that when you go to humans, there are no mismatch risks when implanting the first humans. And of course, then training properly your physician teams to guarantee physician adherence to the protocol and also safety of the patients. We like to say that preclinical science is not a commodity service. You should invest energy and time to make sure that you are successful in this preclinical phase, because otherwise you won't have the green light from the agency to go to human use and to the market. When selecting a preclinical partner, we like to highlight the following. Of course, highest ethical standards have to be met. You have to have a partner in which you have full confidence that confidentiality will be respected. And here at IMMR, we have full processes in force to respect confidentiality. The scientific independence of the lab is looked at by the agency, and more the agency, for an example, do not like that the manufacturer perform its own preclinical testing. They like that a sub-party, a subcontractant, an independent subcontractant is involved to perform the regulatory studies. Of course, this team has to have a problem-solving orientation and mindset to be able to face the problem that we will encounter and solve them. And the platform on which we will be working together has to be a state-of-the-art, latest cutting-edge technology platform. The regulatory expertise and the QA, the quality assurance environments, has to be there and has to comply to FDA, GLP, OECD, GLP, or NMPA requirements in terms of quality. The surgeons and physicians that will be involved into the preclinical evaluation have to feel like home, and we at IMMR like to say that we are reproducing here the exact same conditions as the one that will be used in humans. So thank you for your attention, and now back to Robert, please.
Thank you, Luc. So just to summarize, whether in vascular intervention or any other disease area, preclinical science provides essential information about product design and performance at the scale of human anatomy, and it's critical for ensuring that human clinical trials can be performed safely, correctly, and with compelling outcomes. So the preclinical phase is not just about checking a box, but it's a crucial and significantly value-enhancing stage of medical technology development. Studies, of course, need to be designed and conducted with appropriate scientific rigor and carefully selected models, and at appropriate facilities with requisite procedural and post-operative experience. And of course, pathology evaluation, while required for regulatory studies, as we have heard, can provide valuable insights all throughout the product development pathway. So please don't hesitate to contact us to discuss your preclinical needs. As an essential service, we are open for business, both in Paris and remotely, wherever you are. Thanks very much for your attention.